Awesome. Okay. Well, first of all, thanks so much, Zoe, for organizing. This has been more work than it looks like, at least looking from the sidelines. And so, yeah, as the title says, I'm going to be talking to you about our efforts, mostly failures and a little bit of successes, trying to modify gene expression in poison frogs. And of course, I am here representing a very large number of people who have been working on this. We have been very fortunate to set up this concerted effort to move poison frogs a little bit more into the molecular model organism side of things. And of course, funded by the EDGE program, which funded this as well. And so I guess what guides me and a good number of the people in our team is understanding how phenotypes actually happen and how evolution actually happens at a mechanistic level. So we usually in behavior or in evolutionary biology study phenotypes as a bit of a given, right? This frog is carrying some tadpoles and it has a very bright green stripe and it also has toxins, but the toxins are molecules. The tadpoles are there because of activities in the father's brain and in the tadpoles' brains, and they're also communicating chemically. And all of this is somehow encoded in the genome and generated through interactions with the environment. So all these mechanisms, I think, are a very important and unexplored link in really understanding how evolution works, behavior works, etc. And this guy went the wrong way. And for the most part, our understanding of these functional mechanisms comes from a handful of species that we call the model organisms. And we were just talking, and I've thought for a while, what really is a model organism? And my only definition is that either it eats trash, it grows on sidewalks, or in the case of Xenopus, it was a pregnancy test before chemical pregnancy tests were invented. And so, of course, these are organisms that we've used a lot to understand how phenotypes happen because we can grow them in the lab. And this ability to grow them in the lab is a desirable feature that started out these systems and generated some infrastructure that becomes itself a desirable feature. And so this loop just moves along, and we know more about Drosophila than we know about entire phyla of animals, just because this loop keeps moving and there's a humongous amount of research in these so-called model organisms. However, in ecology and evolution, we also have our model organisms, and in behavior, we also have our model organisms, which have undergone this same cycle of having some desirable feature that just builds up more and more information, which we often use to understand how phenotypes evolve. And so our motivation is to really connect these two, to move stuff from the bench model organism world to the organismal model system world and back again, so that we can actually understand how the mechanisms that generate phenotypes evolve to generate phenotypic evolution. So the model is, of course, poison frogs, which are the coolest frogs, and that is just a given. And something that attracts me about poison frogs, besides their coolness, is that if you look at the phylogeny of poison frogs, you actually find a bunch of different origins of these pretty conspicuous colors associated with toxicity and sometimes with diet specialization and some other behaviors. If you were wondering what the non-colorful looks like, it's that column in the corner. That's actually 4 different genera separated by 30 million years, and taxonomists still have no idea how to really tell them apart. So this is a system with a lot of convergent evolution, which makes comparisons across at least semi-independent events a useful way to say general things. In behavioral terms, since we're at a behavior meeting, poison frogs have a bunch of cool behaviors from parental care, courtship. They fight like crazy. They seem to have super cool spatial abilities that definitely trick researchers. I don't personally work on this, but there's a few people in this room who are actually the world experts on these things, so please ask them about behavior. Poison frogs are also toxic, and the studies of their toxins have been a driving force over 60 years of the research on these frogs. the interactions of the toxins with the frogs themselves, the interactions of the toxins with their predators, the bioperspectives uses of these toxins are all things that we can study mechanistically from a functional point of view to understand the evolution of these organisms as a whole thing. And so one of the people in our team, Lauren, drew up this nice figure to kind of show that the poison frog community studies a bunch of different aspects of these animals: ecology, evolution, behavior, etc. And so they are a very nice system for really doing integrative biology that can allow us to understand how this one clade became what it became. And so our goal here is, of course, moving stuff from, as I said, from the more molecular model organisms to our particular model system. And we focus a lot on sandalfish and zebrafish because of A, phylogenetic, B, just physiological and organismal similarities. And so what we're doing, and don't really pay attention to the details here, is trying to basically generate tools that allow us to do three things. First, as with anything, we need to know what the genes are, how they look like, what their sequences are. So we're putting a lot of effort into generating genomic resources that can be genome assemblies, transcriptomes, per tissues, etc. Everything you find in these nice databases. We actually have our own database, which is called Amphibase, which is very young and small, but hopefully will grow. And we're also looking to do basically what Zoe just explained, either editing the genome or modifying gene expression without actually changing the sequence of the genome. And so what I'm gonna talk about today is our attempts to do these latter two things. The sequencing we can talk about later, but it's not too functional, it's just sequencing. It has its own challenges, but for the purposes of this workshop, I think the two latter are what you're all interested in. And so genome editing usually just consists of getting your gene, designing some way to modify it, which in our case, we mostly use CRISPR. So you design your guide RNAs, etc. You get some embryos, you inject them, you rear them, and then they grow up and show you some nice phenotypes, and you can do experiments, etc. And if you were working on xenopus, you would just start by injecting a female frog who would ovulate and give you 10,000 eggs that you could in vitro fertilize and get enough eggs for a lifetime that all were fertilized exactly at the same time. And so you can basically inject until it's lunchtime and you're good. Poison frogs have complex courtship. And so we need to wait for them to mate. And we need to wait for them to lay eggs. And we can give them some little hiding spots. And sadly, this video isn't gonna work. But you have to believe me, there are some eggs in this little cup that we need to literally just pour out into this petri dish, and it's about 8 eggs. Sadly, you can't see me doing that, but. And so the first thing that we're doing is actually trying to find ways to do this a little more predictably, right? Waiting for frogs to lay eggs when you are around to catch them early enough is perhaps not the most efficient way. And so Maya Gaines Richardson has been working for a while now trying to figure out what type of hormone injection strategies work to A, promote ovulation in females, but B, also make males understand that the females are ovulating and they need to fertilize the eggs. Because since it's so few eggs, doing in vitro fertilization would require an endless supply of males because you need one male for every about 10 eggs. You can make that, do the math. It's not sustainable to kill one male for 10 eggs. And there has been some success, but really we are still mostly relying on stocking the frogs until they lay eggs. But let's say we got some eggs, and this is a nice clutch of eggs. It's relatively young, and you can tell because it's not all dirty and hasn't been taken care of by its dad, who will moisten it and put a bunch of mud on top of it, which makes injection harder. Here's where your problems start, because you can see the coat of jelly in these eggs is non-trivial, right? We need to remove that. We're injecting it with a very thin glass needle going through the thickest jelly you might imagine, which protects these eggs from desiccation in the wild. Is just unfeasible. The jelly is so thick that the Xenopus and salamander protocols don't work to remove this jelly chemically. So you have to do it by hand, which means you kill a bunch of your embryos for the first months. But after that, we can inject them and it actually works really well. A colleague said DNA is DNA and Cas9 cuts it. And that's what happens. Once we manage to get into the egg and not kill it with the injection, we just rear them in these little Petri dishes, changing their substrate about once a day. And they actually grow up. And you can see here, we are now reliably making knockouts of this particular gene that is involved in melanogenesis. And it doesn't show great, but if you compare this tadpole with these three, you can see they have a lot less melanin. And you will see in a second a nicer picture. But if we sequence the bit of the gene that we were trying to cut, you can see this bottom panel that is covered by the closed caption has some very nice peaks that tell you the sequencer is reading one base that is the same every time. Here it is very messy because we have cut the alleles, and so a bunch of the different copies of DNA that we're sequencing have different sequences, and so the sequencer gets confused. If we let these tadpoles grow, oh, come on. That's what the juveniles look like. So clearly we're being able to knock out melanin production. However, this guy was injected when it was a single cell, so we expected it to be a homogeneous mutant, but you can see there are bits that have melanin. And we think this is because the eggs are so big that actually hitting the first nucleus is nearly impossible. You inject a few nanoliters into an egg that is about 1 millimeter across. And so we think the CRISPR chemicals are just hanging out in the egg for a long time and cutting different cell lineages. So we're not sure if this is actually hitting the germline and whether we'll be able to create lines, but with enough editing, we're pretty sure you will eventually get a few that will pass this on to next generations. So we're polarized in terms of yes. So, yeah, so they are polarized, and you can see, so for the non-frog people, before the first cell division, there is this little gray thing called the gray crescent on the egg, which tells you where some of the morphogens and the nucleus should be. But we inject on the order of 2 nanoliters into a big egg. So I don't think we could reliably hit the germline every time, but I think if we inject enough, we should be able to get germline. That being said, we're modifying phenotypes, but in the interest of time, I'll keep going. Now, towards the future, of course, we just don't want to be able to break proteins. And so we're working to develop other CRISPR-mediated techniques, many of which Zoe talked about, and the one that I particularly am very interested in is inserting pieces of DNA that code for RNAi cassettes that will turn off other genes, because this allows us to just into the cellular environment pump out stuff that turns off your gene of interest. And so you can, for example, inject this, put this into genes that are expressed in your tissue of interest or at your developmental time of interest, or maybe co-expressed with your gene of interest. So this is a really powerful tool, I think, to modulate gene expression at a much finer scale. My undergrad, Chris Talbot, is starting to work on this, but everything that survives is not modified. So trial and error, as we've been hearing, is the main subject here. However, there are times when we either don't want to or really can't edit the genome, right? If we want to target a gene that is very important at the early stages, so changing expression kills your embryos, etc. And so the second big axis of our team effort, oh, and I should mention you can sometimes do that in frogs by injecting specific cells in the individual divisions. You can read that paper about how to do it. But if you really can't, then you can just play with different parts of the DNA to protein continuum, right? We have been talking about how you edit DNA, but you can use microRNAs, etc., to modify these other parts of it. And so to do that, we're working on ways to put exogenous material into our tissues of interest in order to modify gene expression there. This is mostly led by Jesse Delia and Maya Gaines Richardson, who I told you about. And so the technique we chose to try to pour it into poison frogs is electroporation, where you hit a biological membrane with electricity, and this opens pores that allow you to sneak stuff into the cell. And so to do this, Jesse and Maya have actually developed a really nice protocol that works on tadpoles of 5 different species across frog families. So this is pretty awesome. The first thing you do is, of course, anesthetize your little tadpole, because you don't want to be mean to it and stab it with a big needle while it's awake. And then you take the tadpole out of the water. And for example, in this case, this needle is injecting a plasmid into the tadpole's brain. And the tadpoles are big enough, but also cartilaginous enough so that this is easy to do and they heal with some practice. And then after you inject, you can actually just zap the whole tadpole with a very mild electric current. And since tadpoles are pretty much wet blobs of tissue, this actually does very efficiently port the cargo into the brain or the muscles. We've done it with both tissues. And it actually works surprisingly well. And I don't mean surprisingly in a way that underappreciates Jesse and Maya's huge efforts to do this. But you can see here in Xenopus, in a glass frog, and in 2 poison frogs, they were able to insert GFP, which is just a shiny green protein, into tail muscles of these tadpoles. And they've, of course, done a very big effort in optimizing the protocols that work very well for each species. This is hopefully coming out published soon. It's in some part of the pipeline. They've also done it in the brain, as I was telling you. And so this seems to be a really promising way to insert plasmids into the brain that express some gene of interest that should be pretty flexible. The setup, as you saw, is really pretty improvisable. So this should be portable to the field. And now that this is done, Sarah Ludington and Najma Akbari have been starting to work on electroporating other stuff that might help regulate gene expression in other ways. This particular photo is of morpholinos, which are these tiny pieces of DNA that act similar to interference RNAs in that they don't let messenger RNA become proteins. You can read up more about them. And this image on the left, you will have to believe me, is a piece of brain where these morpholinos are actually acting. This was sent to me like 4 days ago, so we know we can get these things into the brain. Whatever they're doing is still a mystery, or maybe they know and haven't told me yet. But so this seems to be a very encouraging way that is moving really fast for a non-model system in its development. And so what we've learned over these couple of years, or more than a couple now, of trying to develop functional genomics in poison frogs is that it can definitely be done. And that, I guess, to me, as someone who trained in herpetology, it was surprising that the difficult bit is actually the frogs, not the molecular biology. These genome editing, gene expression editing techniques have been developed at the most common, I guess, feature of life, which is nucleic acids. And so I think we've learned that even though we're the frog experts, we really, really need to learn a lot about frogs to make this work. And of course, having a big community with diverse expertise has been key for this, because I could never do half of the molecular biology. With that, this should go to a thank you slide, but it didn't. So I'm happy to take any clarifying questions. Yeah.
Injections to localize the plasmid to the germplasm, transport it
That's actually a really good idea. I hadn't thought about it, but it definitely deserves exploration. Where the most trouble we're having is getting embryos to survive, partly because we have to maul them pretty badly to get the jelly off before the first cell divisions. But that is a fantastic idea, actually.
Hi, I also studied personal tackle, so this is good. So my question is, do it matter what stage the tackle was for the session?
For the
So like the stage of the tackle, like in talking about.
for the preparation?
Yeah, so just like the 5 minute stuff.
Right. So we haven't tried too many stages. We were doing stages 25 to 28, which to the non-frog people is after they hatch, before they have feet. And that is because they're tiny and their skull is pretty soft. I would expect it to matter. The later you go, the more stuff you can break, the less it can heal, but we haven't tried it.
Well, I would have thought to the opposite of like moving your cardio then, or I've done stuff with the tails, just nothing like you might need, it's for the bigger ones, the better, so.
Okay. That's good to know. Thanks.